Hello again, and welcome. I am so glad that you have chosen to join us for this quiet exploration. Tonight, we will be turning our attention away from the human form and toward another remarkable example of biological design, an animal that shares our homes and yet remains a vessel of ancient predatory perfection. To understand the physiology of the feline is to appreciate a masterpiece of evolution, a living blueprint for efficiency, stealth, and survival. This journey into the inner workings of the cat is in many ways an investment in our appreciation for the natural world itself. The more familiar we become with the intricate machinery that governs another creature's existence, the more profound our respect for the silent, complex processes of life becomes. This knowledge allows us to look upon a simple house cat, curled in a patch of sunlight, and see not just a companion but a highly specialized organism whose every cell and sinew is a testament to millions of years of refinement. As we proceed, we will find that the principles of physiology, of form-meeting function, are universal, yet their expression in the cat is uniquely and quietly spectacular. Our discussion will navigate through the various systems that compose the feline body, from the overarching evolutionary strategy that defines its place in the world, down to the microscopic cells and tissues that serve as its fundamental building blocks. We will proceed slowly, methodically, allowing the structure and order of this biological information to provide a calm and steady focus. There is no need to memorize, only to listen as we uncover the quiet elegance of the cat's internal world. The narrative of feline evolution is written not in historical texts, but in the very blueprint of its biology. To begin our understanding, we must first place this animal within the grand hierarchical system of biological classification, a framework that clarifies its relationship to all other living things. Within the kingdom Mammalia and the phylum Chordata, the cat belongs to the class Mammalia, sharing fundamental traits with a vast array of other warm-blooded, hair-covered vertebrates. Further refinement places it in the order Carnivora, a group defined by its adaptation for meat consumption. It is within the family Felidae, however, that the cat's most specialized identity truly comes into focus. This family, which includes everything from the smallest sand cat to the largest Siberian tiger, represents a lineage of hunters refined to an extraordinary degree. This evolutionary trajectory has culminated in what is formally classified as a hypercarnivore, a designation for organisms whose diet consists of more than 70% meat. This is not merely a dietary preference, but a profound biological commitment, a commitment that dictates nearly every aspect of their internal and external physiology. The relentless evolutionary pressure to become a more effective hunter of live prey has sculpted the feline form over millennia. The demand for stealth has shaped its musculoskeletal system for silent movement. The need for a swift, decisive kill has honed its sensory and neural pathways for rapid response, and the imperative of energy efficiency has optimized its metabolism for a lifestyle of patient waiting, punctuated by explosive action. Every system we will explore in the coming hours is, in some fundamental way, a solution to the complex problems posed by this hypercarnivorous niche. This overarching principle serves as the organizing theme for our entire discussion, the lens through which we can most clearly view and understand the intricate details of feline anatomy and physiology. It is the whys behind the what, providing a quiet, logical context for the remarkable biological machinery we are about to examine. To truly appreciate this external form, however, we must first turn our attention inward to the fundamental units from which this complex organism is constructed. The journey into feline physiology begins, as all biological journeys must, at the cellular level. The cell is the elemental unit of life, a self-contained microscopic world of organized complexity. The feline body is an integrated society of trillions of these cells, each one performing a specific role, yet all working in concert to sustain the whole. A typical feline cell, like that of other mammals, is a eukaryotic cell, characterized by the presence of a membrane-bound nucleus, which houses the animal's genetic material, the DNA that serves as the blueprint for its entire structure and function. Within the cytoplasm, the gelatinous substance filling the cell, lie numerous organelles, each a specialized workshop tasked with a particular aspect of cellular life. The mitochondria, often called the powerhouses of the cell, are responsible for cellular respiration, the process of converting nutrients from the cat's carnivorous diet into adenosine triphosphate, or ATP, the universal energy currency that fuels everything from a muscle twitch to a neural impulse. The ribosomes are the sites of protein synthesis, translating genetic code into the structural and functional proteins that are essential for life. The endoplasmic reticulum and the Golgi apparatus work in tandem as a manufacturing and distribution network, modifying, sorting, and packaging these proteins for use within the cell or for export to other parts of the body. Encasing this entire intricate operation is the plasma membrane, a semi-permeable lipid bilayer that regulates the passage of substances into and out of the cell, maintaining the precise internal environment necessary for its survival. While the single cell is the fundamental unit, life on the scale of a cat requires a higher level of organization. Cells with similar structures and functions group together to form tissues, which are the very fabric of the body. In the feline, as in all vertebrates, we can identify 4 primary types of tissue, each with its own unique characteristics and responsibilities. The first of these is epithelial tissue. This tissue serves as a covering and a lining, forming the outer layer of the skin, the epidermis, which protects the body from the external environment. It also lines the internal passageways and cavities, such as the entire length of the digestive tract, the respiratory airways, and the blood vessels. Epithelial cells are tightly packed, forming a continuous sheet that acts as a selective barrier. They can be squamous or flattened, cuboidal or cube-shaped, or columnar, tall and column-like, with their shape directly relating to their function, be it protection, absorption, or secretion. The second primary type is connective tissue, which, as its name implies, serves to support, connect, and separate other tissues and organs. Unlike the tightly packed cells of epithelium, connective tissue cells are often scattered within an extracellular matrix. This matrix, composed of protein fibers and a fluid or solid ground substance, is what gives each type of connective tissue its characteristic properties. In the feline, this category is remarkably diverse. It includes bone, a rigid connective tissue whose matrix is hardened with calcium salts, providing the structural framework of the skeleton. It includes cartilage, which provides flexible support in areas like the joints and the external ear. It includes dense connective tissue, rich in strong collagen fibers, which forms the tendons that connect muscle to bone and the ligaments that connect bone to bone. Blood, too, is considered a specialized connective tissue, with its cells suspended in a fluid matrix called plasma, serving to transport nutrients, gases, and waste products throughout the body. The third category is muscle tissue, a tissue uniquely specialized for contraction, thereby producing movement. The cells of muscle tissue, known as muscle fibers, contain filaments of the proteins actin and myosin, which slide past one another to generate force. There are 3 types of muscle tissue found in the feline body. Skeletal muscle is attached to the bones of the skeleton and is under voluntary control. It is this tissue that allows for the powerful, deliberate movements of hunting and locomotion, the silent stalk, the explosive pounce, and the rapid pursuit. Under a microscope, skeletal muscle appears striated or striped due to the highly organized arrangement of its contractile filaments. Smooth muscle, in contrast, is found in the walls of internal organs such as the stomach, intestines, and blood vessels. Its contractions are involuntary, responsible for the slow rhythmic processes of digestion and the regulation of blood pressure. Finally, cardiac muscle is found exclusively in the walls of the heart. It is also striated, but its contractions are involuntary, generating the ceaseless rhythmic beat that pumps blood throughout the animal's life. The fourth and final primary tissue is nervous tissue, the body's main communication and control network. This tissue is composed of two main cell types, neurons and glial cells. Neurons are the primary signaling cells, specialized to generate and transmit electrical impulses, known as action potentials, over sometimes considerable distances. They form a complex network that connects all parts of the body to the central control centers, the brain and spinal cord. Glial cells, which are far more numerous, provide support, insulation, and nourishment for the neurons, ensuring the entire system functions efficiently. It is this tissue that receives sensory input from the cat's highly attuned eyes, ears, and whiskers, integrates this information in the brain, and issues motor commands to the muscles, enabling the swift, coordinated responses for which the feline is so well known. Together, these four tissues, epithelial, connective, muscle, and nervous, interweave and cooperate to form the organs and organ systems that constitute the complete functioning animal. This intricate assembly of cells and tissues does not operate in a vacuum. Rather, it exists within a precisely regulated internal environment, a state of dynamic equilibrium known as homeostasis. Homeostasis is one of the most fundamental principles of physiology, referring to the body's ability to maintain relatively stable internal conditions, even when the external world is in a state of constant flux. For an animal like the cat, a predator that must cope with varying environmental temperatures, fluctuating food availability, and the physiological stresses of the hunt, the maintenance of this internal balance is absolutely critical for survival. This regulation is not a static state, but an ongoing active process, a continuous series of adjustments managed by the coordinated efforts of multiple organ systems. One of the most vital homeostatic functions is thermoregulation, the maintenance of a constant internal body temperature. As a mammal, the cat is an endotherm, meaning it generates its own body heat metabolically. This allows it to remain active across a wider range of ambient temperatures than a cold-blooded animal could. However, this ability comes at a metabolic cost and requires sophisticated regulatory mechanisms. When the external temperature drops, the feline body initiates several responses to conserve heat and generate more. Tiny muscles attached to the base of each hair, the erector pili muscles, contract, causing the fur to stand on end. This process, known as pilierection, traps a layer of air close to the skin, which acts as an effective insulator. Blood vessels near the surface of the skin constrict, a process called vasoconstriction, which reduces blood flow to the periphery and minimizes heat loss to the environment. Behaviorally, the cat will seek shelter, curl into a tight ball to reduce its surface area, and may shiver, an involuntary contraction of skeletal muscles that generates significant heat. Conversely, when the body temperature begins to rise, an opposing set of mechanisms is activated. The peripheral blood vessels dilate or widen in a process called vasodilation. This increases blood flow to the skin, allowing more heat to radiate away from the body. While cats have sweat glands, they are primarily located on their hairless paw pads and are not a major contributor to cooling. Instead, a primary method of evaporative cooling is panting. The rapid, shallow breathing increases airflow over the moist surfaces of the tongue and respiratory tract, promoting evaporation and thus dissipating heat. Behaviorally, the cat will seek out cool, shaded areas, stretch out its body to maximize its surface area, and reduce its overall activity level to minimize metabolic heat production. Another critical aspect of feline homeostasis is the regulation of water balance, or osmoregulation. The ancestors of the domestic cat evolved in arid desert environments, and this legacy is evident in their remarkable ability to conserve water. This adaptation is largely a function of their highly efficient kidneys. The kidneys play a central role in osmoregulation by filtering waste products from the blood and adjusting the volume and concentration of urine. Feline kidneys are exceptionally adept at reabsorbing water from the filtrate, allowing them to produce highly concentrated urine. This minimizes water loss and enables them to thrive in conditions where drinking water may be scarce. This renal efficiency is also linked to their naturally low thirst drive. They are able to derive a significant portion of their required water intake directly from the moisture content of their prey. This is a crucial physiological trait that underscores their identity as hypercarnivores, as a diet of fresh meat is inherently rich in water. Finally, the maintenance of homeostasis extends to the regulation of energy and metabolism. The feline lifestyle is one of feast and famine, characterized by long periods of rest and energy conservation, punctuated by the intense energy-demanding activities of hunting. Their physiology is finely tuned to this rhythm. During periods of rest, which can occupy a significant portion of their day, their metabolic rate decreases, conserving the energy gained from their last meal. Their metabolism is specifically adapted to a high-protein, high-fat diet. They utilize amino acids from protein not only for building their own tissues, but also as a primary source of energy through a process called gluconeogenesis, the synthesis of glucose from non-carbohydrate sources. Their ability to efficiently metabolize fats provides a dense, long-lasting energy reserve. This entire system, thermoregulation, osmoregulation, and metabolic regulation, forms a quiet background symphony of constant adjustment, ensuring that the cat's internal environment remains within the narrow range of parameters necessary for life, allowing the animal to maintain its predatory readiness at all times. To support the dynamic processes of life and movement, a structural framework is required, a system that provides both rigidity and the potential for motion. This brings us to the skeletal system of the cat, an architectural marvel that is both lightweight and remarkably strong. The feline skeleton is composed of approximately 244 bones, a number that can vary slightly depending on the length of the tail. This intricate assembly serves multiple critical functions. First and foremost, it provides the fundamental structure and support for the body, giving it its characteristic shape and preventing the collapse of soft tissues. It also serves a protective role, encasing and shielding vital organs from physical trauma. The cranium, for instance, forms a durable vault for the delicate brain, while the rib cage guards the heart and lungs. Furthermore, the skeleton acts as a lever system, providing attachment points for the muscles that generate movement. It is also a site of mineral storage, acting as a reservoir for calcium and phosphorus, which are essential for numerous physiological processes, including muscle contraction and nerve signaling. Finally, within the marrow of certain bones, the crucial process of hematopoiesis, the formation of blood cells, takes place. We can divide the feline skeleton into two main subdivisions for the purpose of orderly examination: the axial skeleton and the appendicular skeleton. The axial skeleton forms the central longitudinal axis of the body. It includes the skull, the vertebral column, the ribs, and the sternum. The skull itself is a complex structure, composed of numerous fused and interlocking bones. It houses the brain, the primary sense organs, and the powerful jaws, which are equipped with the specialized dentition of a carnivore. The vertebral column, or spine, is a defining feature of feline agility. It is comprised of a series of individual vertebrae, extending from the base of the skull to the tip of the tail, categorized into cervical, thoracic, lumbar, sacral, and caudal regions. The exceptional flexibility of the feline spine, particularly in the thoracic and lumbar regions, is due to the unique shape of the vertebrae and the elasticity of the intervertebral discs that cushion them. This flexibility is what allows for the cat's remarkable ability to twist, turn, and absorb the impact of high jumps. The rib cage is formed by the ribs, which articulate with the thoracic vertebrae, and the sternum, or breastbone. It creates a protective yet flexible thoracic cavity, allowing for the expansion and contraction of the lungs during respiration. The appendicular skeleton consists of the bones of the limbs, the forelimbs and hind limbs, and the girdles that attach them to the axial skeleton. The pectoral girdle, which attaches the forelimbs, is notable in the cat for its greatly reduced or absent clavicle or collarbone. This bone is not fused to the rest of the skeleton, but is instead embedded in muscle. This anatomical feature contributes significantly to the cat's narrow build and the fluidity of its stride, allowing the scapula, or shoulder blades, to move with a greater range of motion. This enables a longer, more efficient gait and the ability to squeeze through narrow openings. The forelimbs themselves are adapted for stealth, balance, and the manipulation of prey, consisting of the humerus in the upper arm, the radius and ulna in the forearm, and the complex assembly of carpal, metacarpal, and phalangeal bones that form the wrist and paw. The pelvic girdle is a more robust structure, firmly fused to the sacral vertebrae of the spine. It provides a strong foundation for the powerful hind limbs, which are the primary drivers of locomotion and the source of the cat's explosive leaping ability. The hind limbs are composed of the femur in the thigh, the tibia and fibula in the lower leg, and the tarsal, metatarsal, and phalangeal bones of the ankle and hind paw. The entire skeletal system, in its lightness, strength, and unique articulations, is a silent testament to the evolutionary pressures that have shaped the cat into a paragon of predatory grace. This elegant skeletal framework, however, is merely a passive scaffold. To animate it, to produce the silent stalk and the lightning-fast pounce, requires an active dynamic system. This brings our focus to the sensory organs, the highly sophisticated instruments through which the cat perceives its world and gathers the information necessary to orchestrate its movements. The feline sensory apparatus is finely tuned to the demands of a crepuscular and nocturnal hunter, an animal that operates most effectively in the low-light conditions of dawn and dusk. Its perception of reality is profoundly different from our own, dominated by sight, sound, and touch in ways that are specifically adapted for detecting and tracking prey. The most prominent of these sensory systems is vision. The eyes of a cat are disproportionately large relative to the size of its skull, a clear indication of their importance. Their forward-facing placement provides excellent binocular vision, which allows for precise depth perception, an essential ability for judging distances when leaping or pouncing on a target. The structure of the feline eye contains several key adaptations for superior night vision. The cornea, the transparent outer layer, and the lens are both large and highly curved, allowing them to gather the maximum amount of available light. The pupil, the opening that allows light to enter the eye, is not circular as in humans, but is a vertical slit. This shape allows for a much greater degree of control over the amount of light entering the eye. In bright light, it can constrict to a tiny sliver to protect the sensitive retina, while in dim light, it can dilate to become almost perfectly round, maximizing light-gathering capacity. Behind the lens lies the retina, the light-sensitive tissue that lines the back of the eye. The feline retina is particularly rich in one type of photoreceptor cell, known as rods. Rods are extremely sensitive to low levels of light and are primarily responsible for vision in dim conditions, though they do not perceive color well. The high density of these cells is a primary reason for the cat's excellent night vision. Furthermore, behind the retina lies a specialized reflective layer called the tapetum lucidum. This structure, whose name means bright carpet, acts like a mirror. Any light that passes through the retina without being absorbed by a photoreceptor is reflected back by the tapetum lucidum, giving the rods a second chance to detect it. This mechanism effectively amplifies the available light and is responsible for the characteristic A-shine seen when a light is directed at a cat's eyes in the dark. While these adaptations provide superb low-light sensitivity, they come at the cost of visual acuity, or the ability to see fine detail. The feline visual world is likely less sharply focused and less colorful than our own, but for a hunter in the twilight, sensitivity to movement and shape in near darkness is a far more valuable asset. The world of the cat is not only seen, but also heard, with an acuity that far surpasses human capabilities. The feline auditory system is a precision instrument designed to detect the faint, high-frequency sounds produced by small prey, such as the rustling of a mouse in the undergrowth or the ultrasonic squeaks of rodents. This remarkable sensitivity begins with the external ears, or pinnae. The pinnae are large, funnel-shaped structures that can be independently rotated up to 180 degrees. They are controlled by more than 24 individual muscles, allowing the cat to precisely triangulate the source of a sound without moving its head, a crucial advantage when remaining concealed. Sound waves gathered by the pinnae are channeled down the ear canal to the tympanic membrane, or eardrum, causing it to vibrate. These vibrations are then transmitted across the middle ear by a chain of 3 tiny bones, the ossicles, in the inner ear. These mechanical vibrations are transduced into electrical signals within a spiral-shaped, fluid-filled structure known as the cochlea. The cat's cochlea is particularly sensitive to a very broad range of frequencies, extending well into the ultrasonic spectrum, up to 65,000 Hz or more, far beyond the 20,000 Hz limit of human hearing. This allows them to perceive a dimension of the acoustic world that is entirely silent to us. This auditory specialization is not limited to hearing alone. Also housed within the inner ear is the vestibular apparatus, the organ of balance. This system, composed of fluid-filled semicircular canals and otolithic organs, detects movements and changes in the orientation of the head relative to gravity. The information it provides is processed with incredible speed, enabling the cat's famous riding reflex, the innate ability to orient itself during a fall to land on its feet. This rapid neural processing and integration of vestibular input is fundamental to the cat's agility and its seemingly preternatural sense of balance. The cat's perception of its immediate surroundings is further enriched by its highly developed sense of touch, a world perceived not just by the skin, but by a specialized set of tactile instruments. Central to this system are the vibrissae, commonly known as whiskers. These are not ordinary hairs. They are thick, deeply rooted tactile organs, each connected to a dense network of nerves and blood vessels. They are strategically located on the muzzle, above the eyes, on the cheeks, and even on the back of the forelegs. The vibrissae function as highly sensitive air current detectors and proximity sensors. As the cat moves, even the slightest disturbance in the air, such as that created by an obstacle in the dark, causes the whiskers to vibrate. These vibrations are transmitted to the sensory cells at their base, providing the brain with a detailed, three-dimensional map of the immediate environment. This allows the cat to navigate complex spaces in complete darkness without making contact. The whiskers on the muzzle are also approximately the same width as the cat's body, providing a simple and effective tool for gauging whether it can fit through a narrow opening. This tactile sensitivity extends to the paws. The pads on the bottom of a cat's paws, known as the carpal and metacarpal pads, are thick and leathery, but they are also richly supplied with nerve endings, making them highly sensitive mechanoreceptors. They can detect subtle vibrations in the ground, potentially alerting the cat to the approach of other animals. This sensitivity, combined with the silent, cushioned tread they provide, makes the paws perfect tools for stealthy locomotion. The skin itself, across the entire body, contains a variety of receptors that respond to pressure, temperature, and pain, providing a constant stream of information about the animal's interaction with its physical environment. Together, these sensory systems, the light-gathering eyes, the sound-funneling ears, and the spatially aware whiskers and paws create a rich and detailed perceptual world, a world perfectly tailored to the needs of a silent and efficient predator, allowing it to move through its environment with a confidence and grace born of complete sensory awareness. While sight and sound operate over distances, the chemical senses of olfaction and gustation provide a more intimate, molecular understanding of the environment. For the feline, these senses are not merely for the enjoyment of food, but are critical tools for social communication, territorial marking, and the assessment of prey. The sense of smell, or olfaction, is exceptionally well developed, playing a central role in the cat's behavioral and physiological landscape. The primary organ of smell is the olfactory epithelium, a specialized patch of tissue located high up in the nasal cavity. In the domestic cat, this area is significantly larger and more densely packed with receptor cells than in humans, providing a far greater surface area for the detection of airborne odorant molecules. When a cat inhales, air is drawn over the moist, mucus-coated surface of this epithelium. Odorant molecules dissolve in the mucus and bind to specific olfactory receptor proteins located on the cilia of the olfactory neurons. Each neuron is typically specialized to detect a small number of related odorants. The binding of a molecule triggers an electrical signal in the neuron, which is then transmitted directly to the olfactory bulbs of the brain, a pair of structures located at the very front of the cerebrum. The feline brain dedicates a proportionally larger area to processing these olfactory signals than the human brain does, reflecting the heightened importance of this sensory modality. This powerful sense of smell allows a cat to identify other individuals, recognize its own territory, locate food, and detect the presence of predators or potential mates from considerable distances. Beyond this primary olfactory system, the cat possesses a second, distinct chemosensory organ known as the vomeronasal organ, or Jacobson's organ. This structure consists of a pair of fluid-filled sacs located in the roof of the mouth, just behind the upper incisors, with tiny ducts that open into the oral cavity. It is specialized for detecting non-volatile large molecule chemical cues, such as the pheromones left in urine and glandular secretions by other cats. To deliver these molecules to the vomeronasal organ, the cat employs a characteristic behavior known as the phlegmon response. During this response, the cat will curl back its upper lip, wrinkle its nose, and inhale with its mouth slightly open. This action effectively pumps fluid-containing chemical cues from the mouth up through the ducts into the organ. The signals detected here are processed in a separate neural pathway that connects directly to brain regions involved in reproductive and social behaviors, such as the amygdala and hypothalamus. This dual olfactory system provides the cat with two distinct channels of chemical information: a general-purpose system for detecting environmental odors, and a specialized social signaling system that governs its interactions with other felines. In contrast to its highly sophisticated sense of smell, the feline sense of taste, or gustation, is comparatively simple and highly specialized. Taste is detected by gustatory receptor cells, which are clustered together in structures called taste buds. In the cat, these are primarily located on the tip, sides, and back of the tongue. The feline palate is attuned to the chemical profile of meat. Their taste buds are highly sensitive to the amino acids that signal the presence of protein, which is perceived as a savory or umami flavor. They can also detect sour and bitter tastes, which may help them avoid spoiled or toxic food, and they possess receptors for salty flavors. One of the most notable features of feline gustation, however, is what it lacks. Cats have a genetic mutation that renders their receptors for sweetness non-functional. From a physiological standpoint, this makes perfect sense for a hypercarnivore. In their natural diet, there is no nutritional value in seeking out carbohydrates or sugars, so the ability to taste them has been lost through evolution. Their sense of taste is therefore not an instrument of broad culinary exploration, but a focused functional tool for identifying the essential protein-rich foods required for their survival. The information gathered by these highly developed sensory systems must be integrated and acted upon. This brings us to the systems responsible for executing the precise and powerful movements of the feline body. The skeleton provides the framework, and the muscles provide the force, but it is the seamless integration of these two systems, governed by the nervous system, that produces the cat's signature agility. The source of this remarkable flexibility and grace begins, quite literally, with its backbone. The feline vertebral column is a masterpiece of biomechanical engineering. The individual vertebrae, particularly in the thoracic and lumbar regions, have a shape that allows for a greater degree of rotation and flexion than in many other mammals. They are cushioned by exceptionally thick and elastic intervertebral discs, which act as shock absorbers and permit a significant range of motion. This allows the spine to curve dramatically, enabling the cat to arch its back, curl into a tight ball, or extend its body to its full length during a stride. This spinal flexibility is fundamental to the cat's unique running gait. During a full-speed gallop, the spine flexes and extends like a spring, first coiling to bring the hind legs far forward, almost beneath the chin, and then powerfully extending to propel the body forward in a long, bounding stride. This motion effectively adds the entire length of the back to the propulsive power of the hind limbs, dramatically increasing stride length and speed. Complementing this spinal structure is the unique anatomy of the pectoral girdle. The clavicle, or collarbone, in cats, is a small vestigial bone that is not attached to the shoulder blade or the sternum, but instead floats freely, embedded in muscle. This floating clavicle allows the shoulder blades to move more freely and closely together. This contributes to the cat's narrow chest and allows it to pass through any space that its head can fit through. More importantly for locomotion, it allows the forelimbs to swing forward in a longer, smoother arc, further enhancing the efficiency and length of its stride. The limbs themselves are models of specialized function. The hind limbs are long and powerfully muscled, acting as the primary engine for propulsion. The robust bones of the pelvic girdle are fused to the spine, providing a solid, stable base from which the immense forces required for running and jumping can be generated. The femur, tibia, and associated musculature are all oriented to produce maximal forward thrust. The forelimbs, in contrast, are more refined in their function. While they bear weight and contribute to locomotion, they are also adapted for steering, balance, and the crucial task of grappling with and manipulating prey. The structure of the feline paw, or manuus, allows for a degree of supination, the ability to rotate the wrist to turn the paw upward, which is essential for climbing and holding onto struggling animals. The entire musculoskeletal design, from the spring-like spine and floating clavicle to the powerful hind limbs and dexterous forepaws, works in perfect concert to produce a creature capable of moving with an almost liquid grace, transitioning in an instant from silent, patient stillness to explosive, directed violence. The engine that drives this elegant skeletal chassis is the muscular system. Feline musculature is densely packed and highly efficient, characterized by a predominance of a particular type of muscle fiber that is perfectly suited to the life of a predator. To understand this, we must first recognize that skeletal muscle is not a uniform tissue. It is composed of different types of muscle fibers, each with distinct metabolic and contractile properties. The two primary categories are slow twitch, or type I fibers, and fast twitch, or type 2 fibers. Slow twitch fibers are specialized for endurance. They contract relatively slowly, but are highly resistant to fatigue. They are rich in mitochondria and myoglobin, the protein that stores oxygen in muscle, which gives them a reddish appearance. These fibers are ideal for sustained low-intensity activities, like maintaining posture or walking for long periods. Fast twitch fibers, on the other hand, are built for speed and power. They contract rapidly and forcefully, but fatigue much more quickly. They have fewer mitochondria and less myoglobin, appearing paler in color. These fibers are fueled primarily by anaerobic metabolism, which provides bursts of energy without requiring an immediate supply of oxygen. The muscular anatomy of the cat is heavily dominated by these fast twitch or type 2 fibers. This composition is the physiological basis for the cat's characteristic pattern of activity: long periods of rest and energy conservation, followed by brief, explosive bursts of high-intensity action. The powerful muscles of the hindquarters, responsible for leaping and sprinting, are particularly rich in these fibers. This muscular specialization allows the cat to generate the incredible acceleration and power needed to launch an ambush, chase down prey, or leap to a high perch. The trade-off, however, is a lack of stamina. A cat cannot maintain a high-speed chase for a long duration in the way that a cursorial predator, like a wolf, can. Its entire muscular system is optimized for the single, decisive, and energy-intensive act of the kill. The specialization extends to the very structure of the muscle attachments. The tendons, which connect muscle to bone, are exceptionally strong and elastic, allowing them to store and release kinetic energy like biological springs. This is particularly evident in the long tendons of the limbs, which contribute to the efficiency of running and the immense power of the jump. The control of this powerful muscular system is exquisitely precise. Even the largest muscles, like the gluteals and quadriceps of the hind limb, are innervated by a dense network of motor neurons, allowing for fine gradations of force. This allows for both the brute strength of a killing bite and the delicate, precise movements of the paws during grooming or gentle play. The entire system is a perfect marriage of raw power and refined control, a biological engine tuned for a very specific purpose. From the cellular level of fiber type to the macroscopic arrangement of muscle groups, the feline muscular system is the embodiment of explosive potential, held in a state of patient, efficient readiness. The feline form, with its unique skeletal levers and its engine of fast-twitch muscle fibers, is a system designed for the efficient translation of metabolic energy into kinetic force. At the heart of this design is the intricate and powerful musculature that cloaks the skeleton. The muscles of a cat are not simply bundles of contractile tissue. They are arranged in a complex, layered architecture that allows for a remarkable combination of strength, speed, and subtle control. The major muscle groups reflect the primary demands of a predatory lifestyle. The muscles of the neck, such as the sternomastoid and cleidomastoid, are exceptionally well developed. They provide the strength necessary to control the head during a chase, and more critically, to deliver and maintain a powerful, suffocating bite to the neck of prey. These muscles anchor the head to the pectoral girdle, creating a stable platform for the formidable action of the jaws. The muscles of the back, particularly the long isthmus dorsi and multifidus spini, are long, powerful bands that run along the length of the vertebral column. They work in concert with the abdominal muscles, such as the rectus abdominis and the obliques, to produce the profound flexion and extension of the spine that is so characteristic of the feline gallop during the propulsive phase of a stride. These dorsal muscles contract powerfully to straighten the back, transmitting the force generated by the hind limbs through the entire length of the body. In the recovery phase, the abdominal muscles contract to pull the hind limbs forward, coiling the spine like a spring to prepare for the next explosive extension. This dynamic interplay between the dorsal and ventral muscle groups is the engine that drives the cat's bounding, high-speed locomotion. The limbs, as we have discussed, are functionally specialized, and their musculature reflects this division of labor. The hind limbs are dominated by large, powerful muscle groups designed for raw power. The gluteal muscles of the hip, the quadriceps femoris group on the front of the thigh, and the hamstring group on the back of the thigh are all exceptionally large. These muscles are responsible for the powerful extension of the hip and knee joints that propels the cat forward and upward. The gastrocnemius muscle, or calf muscle, is also robust, connecting to the heel via the strong Achilles tendon and providing powerful extension of the ankle. This entire assembly makes the hind limb a formidable lever for generating explosive force. The forelimbs, by contrast, possess a musculature that balances strength with a greater degree of control and finesse. The muscles of the shoulder, such as the deltoids and the supraspinatus, provide stability and a wide range of motion. The muscles of the upper foreleg, the biceps brachii and triceps brachii, control the flexion and extension of the elbow. However, it is the muscles of the lower foreleg and paw that demonstrate the most refined adaptation. A complex array of extensor and flexor muscles control the movement of the wrist and the individual digits. This fine motor control is essential not only for climbing, where the cat must be able to securely grip surfaces, but also for the critical task of manipulating and pinning down prey. This intricate muscular system, from the powerful bite of the neck to the explosive thrust of the hindquarters and the grasping dexterity of the paws, is a perfectly integrated machine, with each component optimized for its role in the singular drama of the hunt. One of the most iconic and specialized pieces of this predatory toolkit is the feline claw. The claws are not simply keratinous outgrowths like human fingernails. They are formidable biological weapons, sharp, curved, and seamlessly integrated into the anatomy of the paw. Their most remarkable feature is their retractability. In most members of the Felidae family, with the notable exception of the cheetah, the claws are protractile. This means that in their resting state, they are held withdrawn inside protective sheaths of skin at the tips of the toes. This is an essential adaptation that serves two primary purposes. First, it keeps the claws from making contact with the ground during normal locomotion. This prevents them from becoming blunted or worn down, ensuring they remain razor sharp for when they are needed. Second, it allows for a silent, padded tread, a crucial element of a stealthy approach. The mechanism of claw, protraction, and retraction is a marvel of biomechanical efficiency. The final bone of each toe, the distal phalanx to which the claw is attached, is uniquely shaped. In the retracted state, this bone is held in a dorsiflexed or upwardly rotated position by a strong, elastic dorsal ligament. This ligament is constantly under tension, acting like a rubber band to passively hold the claw inside its sheath. This means that retraction is the default, resting state, requiring no active muscle contraction and therefore consuming no energy. To protract the claw, the cat must actively contract the deep digital flexor muscle, which is located in the lower leg. This muscle is connected to the underside of each distal phalanx by a long, strong tendon. When the flexor muscle contracts, it pulls on this tendon, overcoming the tension of the elastic ligament and rotating the distal phalanx downward and forward. This action swings the claw out of its sheath, extending it into its fully deployed, weaponized position. The instant the flexor muscle relaxes, the passive tension of the dorsal ligament immediately snaps the claw back into its protective, retracted state. This elegant system of opposing passive and active forces allows for the instantaneous deployment and retraction of a formidable arsenal, a perfect example of energy-efficient and highly functional anatomical design. The claws themselves are used for a variety of tasks: for gripping surfaces during climbing, for self-defense, for marking territory by scratching surfaces, and most importantly, for seizing and holding onto prey during the final moments of a capture. The culmination of these individual anatomical and physiological systems, the flexible skeleton, the powerful muscles, the sharp claws, and the acute senses, is expressed in the unified and purposeful act of movement. The study of this movement, in the context of the physical forces that govern it, is known as biomechanics. The biomechanics of feline locomotion reveal a creature that is a master of controlled energy expenditure, capable of a wide range of gaits, each one optimized for a particular purpose, from silent stalking to explosive pursuit. A gait is a specific pattern of limb movements during locomotion. Cats employ several distinct gaits, each with its own characteristic rhythm and footfall pattern. The slowest and most common gait is the walk. During a walk, there are always 2 or 3 paws in contact with the ground at any given time, providing a very stable base of support. The footfall pattern is typically diagonal, with the contralateral forelimb and hind limb moving in unison. However, a key feature of the feline walk, particularly when moving cautiously, is the direct register. This means that the hind paw is placed almost exactly into the footprint, just vacated by the forepaw on the same side. This behavior has 2 significant advantages for a stealthy hunter. It minimizes noise, as the cat only has to be mindful of placing its front paws silently, knowing the rear paws will follow the same safe and quiet path. It also minimizes the disturbance of foliage and undergrowth, leaving a less conspicuous trail. As the cat increases its speed, it transitions to a trot. A trot is a 2-beat diagonal gait, in which diagonal pairs of limbs move forward at the same time with a moment of suspension between each step. The body is supported by 2 feet on the ground at a time, providing less stability than a walk, but allowing for greater speed and efficiency over moderate distances. This is a common gait used for covering ground when the cat is not actively hunting, but is moving with purpose. For even greater speeds, the cat will transition into a canter or a gallop. A canter is a 3-beat gait, while a gallop is a faster 4-beat gait. It is in the gallop that the unique biomechanics of the feline spine become most apparent. The feline gallop is of a type known as a rotary gallop, characterized by a leading forelimb and hind limb. Critically, it incorporates 2 periods of suspension, or moments when all 4 feet are off the ground. The first occurs when the cat is fully contracted, with its limbs gathered underneath its body. The second occurs when the cat is fully extended, stretched out in a long, graceful bound. As we have discussed, the powerful flexion and extension of the spine acts as a 5th limb, storing and releasing elastic energy and dramatically increasing the length of the stride. This spinal motion, combined with the power of the hind limbs and the reach of the forelimbs, allows a cat to achieve remarkable bursts of speed, with domestic cats capable of reaching speeds of nearly 30 miles per hour over short distances. This is a highly energy-intensive mode of locomotion, however, and can only be sustained for a limited time, perfectly aligning with the ambush predator strategy of a short, decisive chase. Beyond these linear movements, the biomechanics of the cat also encompass its extraordinary jumping and climbing abilities. A jump is a testament to the explosive power of the hind limb musculature. Before a jump, a cat will crouch, flexing the joints of its hind limbs to their maximum extent. This action effectively loads the muscles and tendons like springs. The subsequent explosive, simultaneous extension of all the joints in the hind limbs releases this stored energy, propelling the body upward and forward. During the flight phase, the cat uses its tail as a dynamic rudder and counterbalance, making fine adjustments to maintain its orientation and prepare for a precise landing. The flexible spine and sensitive vestibular system allow for remarkable mid-air corrections. Upon landing, the forelimbs are the first to make contact, and the joints flex to absorb the significant impact forces, dissipating the energy smoothly through the muscles and skeleton. This entire sequence, from the cautious walk to the explosive leap, is a fluid demonstration of neuromuscular control and biomechanical perfection, a physical expression of millions of years of evolutionary refinement. The kinetic energy and mechanical force generated by the musculoskeletal system are fueled by a continuous supply of metabolic energy derived from the catabolism of ingested food. This brings our focus to the internal machinery responsible for processing this fuel. The digestive system. The feline digestive tract is a model of carnivorous efficiency, a relatively short and straightforward pathway designed for the rapid breakdown and absorption of a high-protein, high-fat diet. The entire system is a visceral reflection of the hypercarnivorous commitment, stripped of the complexities required to digest plant matter and optimized for the singular task of converting animal tissue into usable energy. The process of digestion begins in the oral cavity, or mouth, a chamber specifically equipped for the initial mechanical phase of this process. The most prominent features within the oral cavity are the teeth. Feline dentition is heterodont, meaning the teeth are differentiated into various forms, each with a specialized function. At the front of the mouth are the small incisors, which are primarily used for delicate tasks such as grasping, grooming, and scraping small bits of meat from bone. Flanking the incisors are the long, sharp, conical canine teeth. These are the primary weapons of the cat, designed for piercing deep into the flesh of prey to sever the spinal cord or major blood vessels, delivering a swift, and fatal bite. Further back in the jaw are the premolars and molars, collectively known as the cheek teeth. In a true carnivore, these teeth are not flat for grinding, as they are in herbivores, but are modified into sharp, blade-like structures. The most specialized of these are the carnassial pair, formed by the fourth upper premolar and the first lower molar. These two teeth occlude, or meet, with a precise shearing action, much like the blades of a pair of scissors. This carnassial shear is used to slice through tough skin, muscle, and sinew, cutting large pieces of meat into smaller, swallowable fragments. The jaw itself is capable of very little side-to-side, or lateral, movement. Its action is almost exclusively a powerful vertical hinge motion, further emphasizing its function as a tool for slicing and crushing, not for grinding. The tongue of the cat is another highly specialized organ. Its surface is covered with numerous sharp, backward-facing papillae made of keratin. These spines give the tongue its characteristic rough, sandpapery texture. They serve several functions. They act as a comb during grooming, efficiently removing loose fur and debris, and in feeding, they function like a rasp, helping to lick meat from bones and to manipulate food within the mouth. The mouth also contains salivary glands, but the saliva of a cat has a different primary function than in omnivores or herbivores. It contains very few digestive enzymes, such as amylase, because the cat's diet contains negligible starch. Instead, feline saliva is primarily composed of mucus, which serves to lubricate the food, binding it into a soft bolus that can be easily swallowed. Once mechanically processed and lubricated, the bolus of food is pushed to the back of the throat by the tongue and enters the pharynx. From here, it is propelled by a wave of involuntary muscular contraction, known as peristalsis, down the esophagus, a simple muscular tube that transports the food from the pharynx to the stomach. The stomach is a J-shaped, highly distensible organ that serves as a temporary storage site and the primary location for the chemical digestion of proteins. The inner lining of the stomach is populated by glands that secrete a highly potent gastric juice. This fluid contains two main components, hydrochloric acid and pepsinogen. The hydrochloric acid creates an extremely acidic environment with a pH of 1 to 2. This intense acidity serves to kill most of the bacteria and pathogens ingested with the raw meat, acting as a crucial first line of defense. It also denatures proteins, causing their complex folded structures to unravel, which exposes their peptide bonds to enzymatic action. The acid also activates pepsinogen, converting it into its active form, pepsin. Pepsin is a powerful protease, an enzyme that begins the chemical breakdown of large protein molecules into smaller polypeptide chains. The muscular walls of the stomach churn and mix the food with these gastric juices, producing a semi-liquid, acidic paste known as chyme. After a period of processing in the stomach, the chyme is gradually released in small increments through the pyloric sphincter into the small intestine. The small intestine is the primary site for the final enzymatic digestion of nutrients, and critically, for their absorption into the bloodstream. Relative to body size, the feline small intestine is significantly shorter than that of an omnivore or herbivore. This reflects the high digestibility of a meat-based diet. The complex, lengthy fermentation chambers needed to break down cellulose and other plant fibers are entirely absent. The small intestine is divided into three sections: the duodenum, the jejunum, and the ileum. In the duodenum, the chyme is mixed with digestive secretions from two accessory organs, the pancreas and the liver. The pancreas secretes a cocktail of powerful enzymes, including trypsin and chymotrypsin, which continue the breakdown of proteins into even smaller peptides and amino acids. It also secretes lipase, which begins the digestion of fats, and bicarbonate, which neutralizes the strong acid from the stomach, creating an alkaline environment optimal for these enzymes to function. The liver produces bile, which is stored and concentrated in the gallbladder. Bile is released into the duodenum in response to the presence of fat. It acts as an emulsifier, breaking down large fat globules into tiny droplets, a process which dramatically increases the surface area available for the lipase enzymes to act upon. As the chyme moves into the jejunum and ileum, the final breakdown of nutrients occurs, and the process of absorption begins. The inner surface of the small intestine is not smooth, but is lined with millions of tiny finger-like projections called villi, which are themselves covered in even smaller projections called microvilli. This architecture creates an immense surface area, maximizing the efficiency of nutrient absorption. The final products of digestion, amino acids from proteins, fatty acids and glycerol from fats, along with vitamins and minerals, are transported across the epithelial cells of the villi and enter the capillaries and lymphatic vessels within. From here, these vital nutrients are carried into the general circulation, ready to be delivered to every cell in the body. Any remaining undigested material, along with water, passes into the large intestine. The primary function of the feline large intestine is the reabsorption of water and electrolytes, compacting the waste material into feces, which are then stored in the rectum before being eliminated from the body. The entire digestive journey, from the carnassial shear to the absorptive villi, is a streamlined and highly effective system for converting the flesh of prey into the substance of the predator itself. The delivery of these absorbed nutrients, along with the vital transport of oxygen and the removal of metabolic waste, is the relentless responsibility of the cardiovascular system. This system is a vast and intricate network, composed of the heart, the blood vessels, and the blood itself, a fluid medium that courses through the body in a continuous closed loop. At the center of this network lies the heart, a remarkable muscular organ that functions as a powerful dual-action pump. The feline heart is a four-chambered structure located in the thoracic cavity between the lungs. It is composed of a specialized type of muscle tissue, cardiac muscle, which possesses the intrinsic ability to contract rhythmically throughout the animal's life without conscious input. The four chambers of the heart are organized into two functionally separate pumps, a right side and a left side, each consisting of an upper atrium and a lower ventricle. The right side of the heart is responsible for pulmonary circulation, the circuit that pumps blood to and from the lungs. Deoxygenated blood, returning from its journey through the body's tissues, enters the right atrium. This blood is dark in color, rich in carbon dioxide and other metabolic wastes. From the right atrium, it passes into the right ventricle, which then contracts forcefully to pump the deoxygenated blood through the pulmonary artery to the lungs. In the lungs, the blood flows through a vast network of tiny capillaries that surround the air sacs or alveoli. Here, gas exchange occurs. Carbon dioxide diffuses out of the blood and is exhaled, while freshly inhaled oxygen diffuses into the blood, binding to the hemoglobin molecules within the red blood cells and turning the blood a bright scarlet red. This newly oxygenated blood then returns from the lungs to the left side of the heart via the pulmonary veins, entering the left atrium. This marks the beginning of the systemic circulation, the circuit that distributes oxygen-rich blood to the rest of the body. From the left atrium, the blood flows into the left ventricle. The left ventricle is the most powerful and muscular of the four chambers, as it must generate enough pressure to propel blood throughout the entire body, from the tip of the nose to the end of the tail. When the left ventricle contracts, it ejects the oxygenated blood into the aorta, the largest artery in the body. The aorta branches into a progressively smaller network of arteries, which carry blood to all the organs and tissues. These arteries eventually branch into microscopic vessels called capillaries. It is within the capillary beds that the essential work of the circulatory system takes place. The walls of the capillaries are extremely thin, often only a single cell thick, which allows for the efficient diffusion of substances between the blood and the surrounding tissue cells. Oxygen and vital nutrients, such as the amino acids and fatty acids absorbed from the digestive system, pass out of the blood and into the cells to fuel their metabolic activities. Simultaneously, waste products, such as carbon dioxide and urea, diffuse from the cells back into the blood to be transported away for disposal. After passing through the capillary beds, the deoxygenated blood is collected into small vessels called venules, which merge to form larger veins. The veins then carry the blood back towards the heart, completing the systemic circuit as they empty into the right atrium. This entire continuous cycle, from heart to body, back to heart, then to lungs and back to heart again, is what sustains the life of every cell. The feline heart is capable of beating at a rapid rate, especially during periods of stress or exertion, ensuring that the muscles and brain receive the increased supply of oxygen they require for peak performance. The conduit for this life-sustaining fluid is the intricate network of blood vessels, which can be categorized into three main types based on their structure and function. Arteries are the vessels that carry blood away from the heart. They have thick, muscular, and elastic walls, a structure that is necessary to withstand the high pressure of the blood being forcefully ejected from the ventricles. The elasticity of the arterial walls allows them to expand with each heartbeat, or systole, and then recoil during the heart's relaxation phase, or diastole. This elastic recoil helps to maintain blood pressure and smooth out the pulsatile flow of blood, ensuring a more continuous delivery to the tissues. As arteries travel further from the heart, they branch into smaller vessels called arterioles. The arterioles have a high proportion of smooth muscle in their walls, which allows them to constrict or dilate. This ability to change their diameter makes them the primary sites of regulation for both blood pressure and the distribution of blood flow to different parts of the body. For instance, during exercise, the arterioles supplying the skeletal muscles will dilate to increase blood flow, while those supplying the digestive system may constrict. Capillaries, as previously mentioned, are the smallest and most numerous of the blood vessels. They form vast, interconnected networks, or beds, that permeate every tissue of the body. Their sheer number creates an enormous total cross-sectional area, which causes the blood to slow down significantly as it flows through them. This slow transit time is critical, as it maximizes the time available for the exchange of gases, nutrients, and wastes between the blood and the tissues. The regulation of blood flow through a capillary bed is controlled by tiny rings of smooth muscle called precapillary sphincters, which can open or close to direct blood to areas with the greatest metabolic need. Veins are the vessels that carry blood back towards the heart. By the time blood reaches the veins, the pressure generated by the heart has greatly dissipated. Consequently, the walls of veins are much thinner and less muscular than those of arteries. Because the pressure is so low, especially in the limbs, venous blood flow relies on other mechanisms to return to the heart against the force of gravity. The contraction of surrounding skeletal muscles during movement squeezes the veins, pushing the blood along. To prevent the backflow of blood between these muscle contractions, many veins, particularly those in the limbs, are equipped with one-way valves. This entire vascular system, from the high-pressure aorta to the low-pressure vena cava, is a dynamic and exquisitely regulated plumbing network, ensuring that every corner of the feline body is continuously supplied with the resources it needs to function. The oxygen carried by the cardiovascular system is acquired through the constant rhythmic process of breathing, which is the primary function of the respiratory system. This system comprises the lungs and a series of passageways that connect them to the outside air, working in concert to facilitate the exchange of gases between the atmosphere and the blood. The path of air begins at the nares, or nostrils, which open into the nasal cavities. Within these cavities, the air is warmed, humidified, and filtered. The surfaces are lined with a mucus-secreting epithelium containing cilia, tiny hair-like structures. This mucus layer traps inhaled dust, pollen, and other particulate matter, and the rhythmic beating of the cilia sweeps this trapped debris towards the pharynx, where it can be swallowed and neutralized by stomach acid. From the nasal cavities, the air passes through the pharynx, a passageway shared with the digestive system, and then into the larynx, or voice box. The larynx is a cartilaginous structure that guards the entrance to the lower respiratory tract. It contains the vocal cords, which can vibrate as air passes over them to produce sound. During swallowing, a flap of cartilage, called the epiglottis, closes over the opening of the larynx, preventing food or liquid from entering the airway. Below the larynx lies the trachea, or windpipe, a flexible tube kept open by C-shaped rings of cartilage. The trachea extends down into the chest, where it divides into two primary bronchi, one entering each lung. Within the lungs, the bronchi continue to branch repeatedly, forming a tree-like structure of progressively smaller airways, secondary bronchi, tertiary bronchi, and finally, microscopic bronchioles. This extensive branching serves to distribute the inhaled air to all regions of the lungs. The terminal bronchioles open into the functional units of the lungs, the alveoli. These are tiny hollow air sacs, arranged in clusters like bunches of grapes. The feline lungs contain millions of these alveoli, which collectively provide an immense surface area for gas exchange. The wall of each alveolus is incredibly thin, consisting of a single layer of squamous epithelial cells. This alveoli wall is in intimate contact with the wall of the surrounding pulmonary capillaries, which is also only a single cell thick. This close proximity of air and blood, separated only by a very thin respiratory membrane, is what allows for the rapid and efficient diffusion of gases. Oxygen from the inhaled air diffuses across this membrane into the blood, while carbon dioxide from the blood diffuses in the opposite direction into the alveoli to be removed from the body during exhalation. The mechanical process of breathing, or ventilation, is driven by the muscles of respiration, primarily the diaphragm and the intercostal muscles between the ribs. The diaphragm is a large, dome-shaped sheet of muscle that separates the thoracic cavity from the abdominal cavity. Inhalation is an active process. The diaphragm contracts and flattens, and the external intercostal muscles contract, lifting the rib cage up and outwards. These actions increase the volume of the thoracic cavity. As the lungs are elastic and adhere to the inside of the chest wall, they expand as well, which causes the pressure inside the lungs to drop below atmospheric pressure, and air rushes in. Exhalation, during quiet breathing, is largely a passive process. The diaphragm and intercostal muscles relax. The natural elastic recoil of the chest wall and lungs causes the thoracic cavity to decrease in volume. The pressure inside the lungs rises above atmospheric pressure, and air is forced out. This simple rhythmic cycle of inhalation and exhalation ensures that the alveoli are continuously ventilated, maintaining the concentration gradients necessary for efficient gas exchange, and keeping the blood constantly supplied with the oxygen required to fuel the predatory machine. A final enigmatic feature of the feline respiratory system is the purr. The exact mechanism of the purr is still a subject of scientific debate, but the leading theory proposes it is produced by the rhythmic cyclical contraction and relaxation of the laryngeal and diaphragmatic muscles at a frequency of 25 to 150 Hz. This creates a regular vibration and pressure change during both inhalation and exhalation, producing the characteristic continuous humming sound. While often associated with contentment, cats also purr when injured or stressed, leading some researchers to hypothesize that the low-frequency vibrations may have a therapeutic self-healing function, promoting bone density and tissue repair. The efficient processing and distribution of nutrients and oxygen are predicated on a diet to which the feline is exquisitely adapted. This brings our focus to the unique metabolic landscape of the cat, a landscape shaped entirely by its evolutionary history as an obligate carnivore. The term obligate, or true carnivore, signifies a biological necessity, not merely a preference. It means that the cat's physiology is so fundamentally adapted to a meat-based diet that it cannot obtain all the essential nutrients it requires for survival from plant matter. This metabolic specialization is evident at the most fundamental biochemical levels and dictates a set of strict, non-negotiable dietary requirements that distinguish the feline from almost all other domesticated animals. One of the cornerstones of this specialization is the cat's unique protein and amino acid metabolism. Unlike omnivores or herbivores, whose bodies can down-regulate the activity of liver enzymes involved in protein catabolism during periods of low protein intake, the cat's liver enzymes are permanently set to a high level of activity. This is an adaptation to a consistently high-protein diet, where amino acids are a constant and primary source of energy, not just building blocks for tissue. This constant, high rate of protein breakdown means that cats have a significantly higher baseline requirement for dietary protein than most other mammals. Furthermore, this metabolic pathway makes them dependent on their diet for several specific amino acids that other species can synthesize themselves. Perhaps the most well-known of these is taurine. Taurine is an amino acid that in most mammals can be synthesized from other sulfur-containing amino acids like methionine and cysteine. However, cats have a very limited ability to perform this synthesis. At the same time, their bodies use taurine for a number of critical physiological functions. It is essential for the formation of bile salts, which are necessary for the proper digestion and absorption of fats. It is also a crucial component for the normal functioning of the retina of the eye and the myocardium, the muscle tissue of the heart. A diet deficient in taurine will over time lead to a series of severe and ultimately fatal health conditions, including feline central retinal degeneration, which results in irreversible blindness, and dilated cardiomyopathy, a condition where the heart muscle weakens and fails. Because taurine is abundant in animal tissues but virtually absent in plant matter, this metabolic dependency firmly locks the cat into its carnivorous niche. Another essential amino acid for the cat is arginine. While arginine is essential for many species, particularly during growth, the feline requirement is exceptionally acute and immediate. Arginine is a key component of the urea cycle, the metabolic pathway that detoxifies ammonia, a toxic byproduct of protein metabolism, by converting it into urea, which can then be safely excreted in the urine. Because of the cat's constitutively high rate of protein catabolism, this cycle is always operating at a high capacity. If a cat consumes a single meal that is deficient in arginine, it cannot produce the necessary intermediates for the urea cycle. Ammonia rapidly builds up in the bloodstream, a condition known as hyperammonemia, which can lead to severe neurological symptoms and death within a matter of hours. Like taurine, arginine is plentiful in animal protein, ensuring this metabolic crisis does not occur in a natural dietary context. The cat's metabolic specialization also extends to its handling of fats and vitamins. As carnivores, they are highly adapted to metabolizing fats for energy, and their diet must contain certain essential fatty acids, such as arachidonic acid, which they cannot synthesize from plant-based precursors. Their vitamin metabolism is similarly specialized. A striking example is vitamin A. Herbivores and omnivores can synthesize vitamin A from beta-carotene, the pigment found in many plants, such as carrots. Cats lack the necessary intestinal enzyme to perform this conversion. They must therefore obtain preformed vitamin A directly from their diet, which is readily available in the liver and fat of their prey. A similar dependency exists for niacin, a B vitamin, and for vitamin D, which they are inefficient at synthesizing in their skin through exposure to sunlight, and must therefore obtain from their food. This entire suite of metabolic particularities, the high protein requirement, the permanent activity of liver enzymes, and the dietary necessity for taurine, arginine, arachidonic acid, and preformed vitamins is not a collection of deficiencies. Rather, it is a highly refined and efficient system of specialization, a metabolic streamlining that has jettisoned the costly and complex biochemical machinery required for processing plant materials, opting instead for a complete and total reliance on the nutrient profile of animal tissue. The intricate symphony of metabolic processes, sensory inputs, and muscular outputs is governed and coordinated by two master control systems, the nervous system and the endocrine system. These systems work in concert to monitor the body's internal and external environments, to process information, and to generate appropriate, coordinated responses, ensuring the animal's survival. We will first turn our attention to the nervous system, the body's high-speed communication network, which is responsible for rapid, targeted control of the body's functions. For the purpose of orderly discussion, we can divide the nervous system into two main structural components: the central nervous system, or CNS, and the peripheral nervous system, or PNS. The central nervous system is the primary integration and command center. It consists of the brain and the spinal cord, both of which are protected by the bony casings of the skull and the vertebral column, respectively. The brain is the seat of all complex neural processing. It can be subdivided into several major regions, each with specialized functions. The cerebrum is the largest part of the feline brain and is divided into two cerebral hemispheres. Its convoluted outer layer, the cerebral cortex, is responsible for higher-order functions, such as sensory perception, the initiation of voluntary movement, learning, and memory. Within the cerebrum are regions dedicated to processing the vast amounts of information received from the cat's highly developed sensory organs. The occipital lobe, for instance, is dedicated to processing visual information, while the temporal lobe processes auditory signals. Beneath the cerebrum lies the cerebellum, a structure that is exceptionally well developed in the cat. The cerebellum does not initiate movement, but rather acts as a coordinator and refiner of motor activity. It receives sensory information about the body's position in space from the eyes, inner ears, and stretch receptors in the muscles, and it compares this information with the motor commands being issued by the cerebrum. It then sends out corrective signals to fine-tune the movements, ensuring they are smooth, balanced, and accurate. The large size and complexity of the feline cerebellum are a direct reflection of the incredible agility and coordination required for a predatory lifestyle, from balancing on a narrow ledge to executing a complex twisting leap. The brainstem, which connects the cerebrum and cerebellum to the spinal cord, controls the most fundamental, automatic functions necessary for life. It regulates heart rate, breathing, blood pressure, and states of consciousness, such as sleeping and waking. It is the primitive, life-sustaining core of the brain. The spinal cord is a long, thin, tubular bundle of nervous tissue that extends from the brainstem down the length of the back. It serves two primary functions. It acts as a pathway for nerve impulses traveling between the brain and the rest of the body, and it serves as a center for coordinating many simple reflexes that do not require conscious input from the brain. The peripheral nervous system consists of the vast network of nerves that extend from the central nervous system, connecting it to the limbs, organs, and sensory receptors throughout the body. These nerves are bundles of axons, the long projections of nerve cells, or neurons. We can functionally divide the peripheral nervous system into two further subdivisions, the somatic nervous system and the autonomic nervous system. The somatic nervous system is responsible for carrying sensory information from the skin, muscles, and joints to the CNS, and for transmitting motor commands from the CNS to the skeletal muscles. It is the system that governs most voluntary conscious movements, such as walking, pouncing, and grooming, as well as processing sensations of touch, pain, and temperature. The autonomic nervous system, in contrast, manages the involuntary functions of the body's internal organs and glands. It operates automatically, without conscious effort, to maintain homeostasis. The autonomic nervous system itself is composed of two branches that generally have opposing effects: the sympathetic division and the parasympathetic division. The sympathetic division is often described as the fight or flight system. It prepares the body for intense physical activity and stressful situations. When activated, it increases heart rate and blood pressure, dilates the pupils, and diverts blood flow away from the digestive system and towards the skeletal muscles, readying the animal for immediate explosive action. This is the system that is dominant during a hunt or a confrontation. The parasympathetic division is known as the rest and digest system. It promotes functions associated with a calm resting state. It slows the heart rate, stimulates the digestive processes, and conserves energy. These two divisions work in a dynamic balance, allowing the cat to transition smoothly between states of high alert and restful conservation, a rhythm that perfectly mirrors its predatory lifestyle. The entire nervous system, from the complex processing of the cerebral cortex to the automatic adjustments of the autonomic nerves, is a highly integrated and incredibly fast network that enables the cat to perceive its world with stunning clarity and to react to it with lethal precision. The ability of the nervous system to perform these remarkable feats of communication and control is dependent upon the function of its fundamental units, the neurons. These specialized cells are designed to transmit information in the form of electrochemical signals. A typical neuron consists of a cell body, which contains the nucleus, a series of branching extensions called dendrites, which receive signals from other neurons, and a single long projection called an axon, which transmits signals away from the cell body to other cells. The junction between the axon of one neuron and the dendrite of another is called a synapse. At the synapse, information is transmitted from one cell to the next, not through direct electrical contact, but through the release of chemical messengers known as neurotransmitters. When an electrical impulse, or action potential, travels down the axon and reaches the synapse, it triggers the release of neurotransmitters into the tiny gap between the two cells. These molecules then diffuse across the gap and bind to specific receptor proteins on the membrane of the receiving neuron. This binding can have one of two effects. It can be excitatory, making the receiving neuron more likely to fire its own action potential, or it can be inhibitory, making it less likely to fire. Each neuron receives thousands of these excitatory and inhibitory inputs from other neurons. It continuously integrates this information, and if the sum of the excitatory signals reaches a certain threshold, the neuron will generate an action potential of its own, propagating the signal further along the neural pathway. This complex process of synaptic transmission and neural integration is the fundamental basis of all information processing in the brain. One of the most classic and elegant demonstrations of the nervous system's function in the cat is the reflex arc. A reflex is a rapid, involuntary, and predictable response to a specific stimulus. Many simple reflexes are mediated entirely at the level of the spinal cord, allowing for an almost instantaneous reaction time because the signal does not have to travel all the way to the brain to be processed. A well-known example is the withdrawal reflex. If a cat steps on a sharp object, sensory receptors in the paw send a pain signal along a sensory neuron to the spinal cord. Within the spinal cord, this sensory neuron synapses directly with an interneuron, a short connecting neuron, which in turn synapses with a motor neuron. This motor neuron then sends an immediate command to the flexor muscles of the leg, causing them to contract and pull the paw away from the painful stimulus. This entire circuit, from stimulus to response, occurs in a fraction of a second, long before the sensation of pain is consciously perceived by the brain. A more complex and uniquely feline example is the righting reflex. This is the innate ability, present even in very young kittens, to orient the body during a fall so that the cat lands on its feet. This is not a simple reflex, but a complex, coordinated sequence of movements, mediated by several parts of the nervous system. The process is initiated by the vestibular apparatus in the inner ear, which detects the change in head orientation relative to gravity. This information is sent to the brainstem, which then orchestrates a rapid sequence of motor commands. First, the muscles of the neck contract to rotate the head back to a normal level position. The rest of the body then follows, twisting in sequence. First, the forelimbs and upper torso, then the hind limbs and lower torso, all aligning themselves with the corrected position of the head. Throughout the fall, the cerebellum is constantly receiving sensory feedback and issuing corrective signals to ensure the movements are smooth and accurate. This remarkable reflex highlights the speed and sophistication of the feline nervous system, a system that can take complex sensory data about motion and gravity and translate it almost instantaneously into a life-saving full-body motor response. This integration of sensory input and motor output, from the simplest spinal reflex to the most complex cortical processing, is what allows the cat to navigate its world with such apparent effortlessness and precision. While the nervous system provides rapid, point-to-point control through electrical impulses and localized neurotransmitter release, the body requires a second, complementary system for slower, more widespread, and longer-lasting regulation. This role is fulfilled by the endocrine system. The endocrine system is a network of ductless glands and tissues that produce and secrete chemical messengers, known as hormones, directly into the bloodstream. These hormones then travel throughout the body, acting on specific target cells that possess the appropriate receptors. In this way, the endocrine system coordinates a vast array of physiological processes, including growth, metabolism, stress responses, and the complex cycles of reproduction, ensuring that the body's internal state is appropriately matched to its developmental stage and environmental conditions. The master control center of the endocrine system is the hypothalamus, a small but vital region located at the base of the brain. The hypothalamus acts as the crucial link between the nervous system and the endocrine system. It receives neural input from many other brain regions, constantly monitoring the body's internal state, including temperature, nutrient levels, and hydration. In response to this information, it produces a variety of releasing and inhibiting hormones that act directly on the pituitary gland, which is suspended just below it. The pituitary gland, often referred to as the master gland, is a small pea-sized structure divided into two distinct lobes. The anterior pituitary. The anterior pituitary produces and secretes a range of trophic hormones, which are hormones that control the activity of other endocrine glands throughout the body. For example, in response to signals from the hypothalamus, the anterior pituitary releases thyroid-stimulating hormone, or TSH, which travels to the thyroid gland and stimulates it to produce its own hormones. It also produces adrenocorticotropic hormone, or ACTH, which acts on the adrenal glands, and the gonadotropins, follicle-stimulating hormone, FSH, and luteinizing hormone, LH, which regulate the function of the ovaries and testes. The posterior pituitary, in contrast, does not produce its own hormones, but instead stores and releases two hormones that are produced in the hypothalamus: antidiuretic hormone, ADH, which is crucial for water balance by regulating the kidneys, and oxytocin, which is involved in social bonding and smooth muscle contraction during birth and lactation. This hierarchical arrangement, with the hypothalamus controlling the pituitary, which in turn controls many other endocrine glands, allows for precise and coordinated regulation of the body's entire hormonal landscape. One of the most important endocrine glands in the cat is the thyroid gland, a butterfly-shaped structure located in the neck, wrapped around the trachea. The thyroid gland produces two key hormones, thyroxine T4 and triiodothyronine T3, which are synthesized using iodine. These thyroid hormones act on nearly every cell in the body to regulate the basal metabolic rate. They essentially set the pace of the body's metabolism, influencing how quickly cells consume oxygen and convert nutrients into energy. In a predator like the cat, the proper regulation of metabolism is critical for maintaining the energy reserves needed for hunting, while conserving energy during periods of rest. The thyroid hormones are also essential for normal growth and development, particularly of the skeletal and nervous systems. Closely associated with the thyroid are the adrenal glands, a pair of small glands located on top of each kidney. Each adrenal gland is composed of two distinct parts, an outer cortex and an inner medulla, which function as separate endocrine organs. The adrenal cortex produces a class of steroid hormones called corticosteroids. These include glucocorticoids, such as cortisol, which play a central role in the stress response by increasing blood glucose levels and suppressing inflammation, and mineralocorticoids, such as aldosterone, which regulates salt and water balance by acting on the kidneys. The adrenal medulla, on the other hand, is functionally an extension of the sympathetic nervous system. In response to a direct neural signal indicating stress or excitement, the fight or flight response, the medulla secretes two catecholamine hormones, epinephrine, also known as adrenaline, and norepinephrine. These hormones are released rapidly into the bloodstream and produce widespread effects that reinforce the actions of the sympathetic nerves. They increase heart rate and contractility, dilate airways, and mobilize energy reserves, preparing the body for immediate, intense physical exertion. This dual-action stress response, involving both the slower, more sustained hormonal action of the cortex and the rapid neurohormonal action of the medulla, provides the cat with a robust and flexible system for coping with the challenges of its environment. The pancreas, an organ we have already discussed in the context of digestion, also serves a critical endocrine function. Scattered throughout the pancreatic tissue are small clusters of endocrine cells known as the islets of Langerhans. These islets produce two hormones that are essential for the regulation of blood glucose levels: insulin and glucagon. After a meal, as nutrients are absorbed and blood glucose levels rise, beta cells in the islets release insulin. Insulin acts on cells throughout the body, particularly in the liver, muscle, and fat tissue, stimulating them to take up glucose from the blood and either use it for immediate energy or store it for later use in the form of glycogen or fat. This action effectively lowers blood glucose levels. Conversely, when blood glucose levels fall, such as during a period of fasting, alpha cells in the islets release glucagon. Glucagon primarily acts on the liver, stimulating it to break down its stored glycogen, a process called glycogenolysis, and to synthesize new glucose from amino acids, gluconeogenesis, releasing this glucose into the bloodstream to raise its concentration. This constant, balanced interplay between insulin and glucagon ensures that the brain and other tissues have a steady supply of the glucose they need to function, a homeostatic mechanism of profound importance. Together, these endocrine glands and the hormones they produce form a silent chemical communication network that orchestrates the slow, deliberate, and fundamental processes of life, from the moment-to-moment regulation of blood sugar to the overarching rhythms of growth and reproduction. The intricate hormonal orchestration of the endocrine system finds its most complex and cyclical expression in the process of reproduction. Feline reproductive physiology is characterized by a set of highly specialized adaptations that are finely tuned to maximize the chances of successful conception and rearing of offspring in a solitary predator. The entire process, from sexual maturation to courtship and gestation, is governed by a complex interplay of hormones released from the hypothalamus, the pituitary gland, and the gonads, the ovaries in the female and the testes in the male. In the male cat, or tom, the testes serve two primary functions: the production of sperm through a process called spermatogenesis, and the synthesis and secretion of the primary male sex hormone, testosterone. Testosterone is a steroid hormone responsible for the development and maintenance of male secondary sexual characteristics, such as the larger head and thicker skin of a mature tom, and for driving male reproductive behaviors, including roaming, territorial marking with urine, and courtship vocalizations. The production of both sperm and testosterone is under the control of the gonadotropins from the anterior pituitary. Follicle-stimulating hormone, FSH, primarily acts on the seminiferous tubules within the testes to stimulate sperm production, while luteinizing hormone, LH, acts on the Leydig cells to stimulate the synthesis of testosterone. This system is maintained in a relatively stable state after the cat reaches puberty, with a continuous production of sperm. The reproductive physiology of the female cat, or queen, is considerably more complex and cyclical. Unlike species that have regular, spontaneous ovulatory cycles, the domestic cat is a seasonally polyestrous induced ovulator. Seasonally polyestrous means that she undergoes multiple reproductive cycles, or estrous cycles, but only during certain times of the year, typically when the length of daylight is increasing in the spring and summer. Induced ovulator refers to a remarkable physiological adaptation wherein the act of mating itself is the trigger that causes the ovaries to release an egg, or ovulate. This mechanism is highly efficient for a solitary species, as it ensures that ovulation occurs only when a viable male is present and mating has successfully occurred, thus conserving the female's metabolic energy and maximizing the probability of fertilization. The estrous cycle of the queen is governed by a fluctuating cascade of hormones. As the cycle begins, Under the influence of increasing day length, the hypothalamus begins to secrete gonadotropin-releasing hormone. This stimulates the anterior pituitary to release FSH. FSH travels to the ovaries and stimulates the growth and development of several ovarian follicles, each of which contains an oocyte, or developing egg. As these follicles mature, they begin to produce the primary female sex hormone, estrogen. The rising levels of estrogen in the bloodstream bring the queen into the behavioral state of estrus, or heat. This is the period of sexual receptivity, characterized by distinct behaviors such as loud vocalizing, restlessness, and adopting a specific receptive posture known as lordosis. This estrogen-driven phase can last for several days, during which the queen actively seeks a mate. If mating does not occur during estrus, the follicles will eventually regress, estrogen levels will fall, and the queen will enter a short period of reproductive quiescence before the cycle begins anew. However, if mating does occur, the physical stimulation of the cervix by the male's penis triggers a neurohormonal reflex. Nerve impulses travel from the cervix to the hypothalamus, causing a massive surge of GnRH to be released. This GnRH surge, in turn, triggers a surge of luteinizing hormone, LH, from the pituitary. It is this LH surge that is the direct trigger for ovulation. Approximately 24 to 50 hours after mating, the mature follicles on the ovaries rupture, releasing their eggs. After ovulation, the remnants of the ruptured follicles transform into a temporary endocrine structure called the corpus luteum. The corpus luteum produces another crucial hormone, progesterone. Progesterone is the hormone of pregnancy. It prepares the uterus for implantation of the fertilized eggs and is responsible for maintaining the pregnancy. If fertilization is successful and the embryos implant in the uterine wall, the corpus luteum will continue to produce progesterone throughout the gestation period, which lasts approximately 63 to 65 days. If fertilization does not occur, or if the embryos fail to implant, the corpus luteum will regress after a few weeks, progesterone levels will drop, and the queen will eventually return to estrus, ready to begin the cycle once more. This intricate, stimulus-driven reproductive strategy is a perfect example of physiological efficiency, a system that tightly links the profound biological investment of ovulation directly to the act that makes it worthwhile. The outward expression of the cat's internal state and its primary interface with the physical world is the integumentary system. This system, composed of the skin, fur, claws, and various glands, is far more than a simple protective covering. It is a complex, multifunctional organ that plays a vital role in thermoregulation, sensory perception, physical protection, and critically, in the subtle and complex language of chemical communication. The skin itself is the largest organ of the body and is composed of two main layers, the outer epidermis and the inner dermis. The epidermis is a stratified squamous epithelium, meaning it is composed of multiple layers of flattened cells. The deepest layer of the epidermis is where new cells are constantly being produced through mitosis. As these new cells form, they push the older cells above them towards the surface. During this upward migration, the cells undergo a process called keratinization, in which they become filled with a tough, fibrous, water-resistant protein called keratin. By the time the cells reach the outermost layer, the stratum corneum, they are no longer living. This layer of dead, keratinized cells provides a durable, flexible, and virtually waterproof barrier that protects the underlying tissues from mechanical injury, dehydration, and invasion by microorganisms. The epidermis is also home to melanocytes, specialized cells that produce the pigment melanin, which gives the skin and fur its color, and provides some protection against the damaging effects of ultraviolet radiation from the sun. Beneath the epidermis lies the dermis, a much thicker and more complex layer, composed of dense, irregular connective tissue. The dermis provides the skin with its strength and elasticity, thanks to a rich matrix of collagen and elastin fibers. It is also densely populated with a wide array of structures. It contains the blood vessels that supply the skin with nutrients and play a key role in thermoregulation by constricting or dilating. It is interwoven with a network of nerve endings and specialized sensory receptors that detect touch, pressure, pain, and temperature. The dermis also houses the hair follicles, the tiny invaginations of the epidermis from which each individual hair grows. Attached to each hair follicle is a tiny smooth muscle, the erector pili, which can contract to make the hair stand on end. Also associated with the hair follicles are the sebaceous glands, which produce an oily, waxy substance called sebum. Sebum is secreted onto the hair and skin, where it helps to lubricate and waterproof the fur, keeping it supple and providing a barrier against moisture. Beyond its role in physical protection, the feline integumentary system is a primary organ of communication, largely through the production and deposition of chemical signals, or pheromones. Cats have a variety of specialized skin glands located in key areas of the body that are used for scent marking. There are sebaceous glands located on the chin, the lips, the temples, and at the base of the tail, as well as interdigital glands located between the paw pads. When a cat rubs its face against an object or a person, a behavior known as bunting, it is depositing facial pheromones from these glands. These pheromones convey a message of familiarity and contentment, marking the object as a safe and recognized part of its territory. Similarly, when a cat scratches an object, it is not only leaving a visual mark and conditioning its claws, it is also depositing scent from the interdigital glands, leaving a clear territorial signal for other cats. This complex use of glandular secretions turns the skin into a dynamic signaling organ, allowing the cat to communicate a rich vocabulary of social and territorial information silently and invisibly, a chemical language that is fundamental to its solitary and territorial nature. The skin is therefore not just a barrier, but a canvas upon which the cat writes its identity and claims its place in the world. The most conspicuous feature of the feline integument is its coat, a dense covering of hair or fur that serves as a primary defense against the elements and as a canvas for a remarkable diversity of colors and patterns. Each individual hair is a non-living structure, composed primarily of keratin that grows from a living follicle embedded in the dermis. The coat is not a uniform covering, but is typically composed of two main types of hair. The longer, thicker hairs are known as guard hairs. These form the outer layer of the coat, providing the primary protection against moisture and physical abrasion, and they are the hairs that display the most prominent color and pattern. Beneath the guard hairs lies a softer, finer, and often denser layer of undercoat, or down hairs. The primary function of the undercoat is insulation. Its fine, often crimped structure is highly effective at trapping a layer of air close to the skin, which significantly reduces the loss of body heat in cold conditions. The relative proportion of guard hairs to undercoat hairs can vary dramatically between different breeds of cat, reflecting their adaptation to different climates. The color and pattern of the feline coat are determined by the presence, type, and distribution of melanin, the same pigment that colors the skin. There are two primary types of melanin: eumelanin, which produces black and brown pigments, and pheomelanin, which produces red and yellow pigments. The vast array of coat colors seen in domestic cats is the result of a complex interplay of multiple genes that control the production and expression of these two pigments. The agouti gene, for instance, controls whether the hair shaft will have bands of different pigment along its length. The dominant form of this gene produces the classic tabby pattern, where each individual hair has alternating bands of light and dark pigment, creating a ticked or patterned appearance. The recessive non-agouti form of the gene results in a solid colored coat, where each hair is a uniform shade from base to tip. Other genes control the density of the pigment granules, which can dilute a black coat to a blue or gray, or a red coat to a cream. Yet other genes are responsible for the classic patterns of the tabby cat, the mackerel, the classic blotched, the spotted, and the ticked, by controlling the distribution of the agouti banded hairs relative to the solid colored hairs. The genetics of coat color in cats is a field of considerable complexity, involving dominant and recessive alleles, sex-linked inheritance, and epistatic interactions where one gene can mask the effect of another. For example, the gene for the white coat is a dominant gene that is epistatic to all other color genes. A cat that inherits this dominant white allele will have an entirely white coat, regardless of what other color or pattern genes it carries in its genotype, because this gene effectively prevents the migration of melanocytes into the skin and hair follicles during embryonic development. Similarly, the gene responsible for the pointed pattern seen in breeds like the Siamese is a form of temperature-sensitive albinism. This gene codes for a version of the enzyme tyrosinase, which is necessary for melanin production, that is only functional at cooler temperatures. As a result, pigment is only produced in the cooler extremities of the body, the ears, face, paws, and tail, while the warmer core of the body remains pale. This intricate genetic tapestry not only produces the aesthetic diversity we observe in domestic cats, but also provides a living record of their selective breeding and evolutionary history. Functionally, these colors and patterns primarily serve the purpose of camouflage, or crypsis, allowing the wild ancestors of the domestic cat to blend seamlessly into their natural environment, a critical advantage for an ambush predator. Beneath this protective and communicative outer layer lies a silent and vigilant internal defense network, the immune system. This system is a complex and diffuse collection of cells, tissues, and molecules that work together to protect the body from a constant barrage of foreign invaders, such as bacteria, viruses, fungi, and parasites. It is a system of surveillance and response, capable of distinguishing self, the body's own cells, from non-self or foreign material, and launching a targeted attack to neutralize and eliminate any potential threats. The immune system's defenses can be broadly categorized into two interconnected branches: the innate immune system and the adaptive immune system. The innate immune system provides the first line of defense. Its responses are nonspecific, meaning it reacts in the same general way to any foreign intruder, and it is always ready to act immediately. The physical barriers of the integumentary system, such as the skin and the mucous membranes, are part of this innate defense, preventing most pathogens from ever entering the body. If these barriers are breached, a second tier of innate defenses is activated. This includes a variety of specialized white blood cells. Phagocytes, such as neutrophils and macrophages, are the scavengers of the immune system. They patrol the tissues, engulfing and digesting any foreign particles, cellular debris, or invading microorganisms they encounter. Another type of cell, the natural killer cell, is specialized to recognize and destroy the body's own cells that have become infected with viruses or have turned cancerous. The innate system also includes the inflammatory response. When tissues are injured or infected, damaged cells release chemical signals, such as histamines. These signals cause local blood vessels to dilate and become more permeable. This increases blood flow to the area, bringing with it an influx of phagocytic cells and plasma proteins to the site of the problem, resulting in the characteristic swelling, redness, and heat of inflammation. This process helps to contain the infection and initiate the process of tissue repair. While the innate immune system provides a rapid general defense, it has no memory of past encounters. This is the role of the adaptive immune system. The adaptive response is highly specific, targeting particular molecular features or antigens of a specific pathogen. Critically, it also possesses immunological memory, which allows it to mount a much faster and stronger response upon subsequent encounters with the same pathogen. The key players in the adaptive immune system are a class of white blood cells known as lymphocytes, which are divided into two main types: T lymphocytes, T cells, and B lymphocytes, B cells. B cells are responsible for antibody-mediated immunity. When a B cell encounters an antigen that matches the specific receptor on its surface, it becomes activated, often with the help of a T cell. The activated B cell then proliferates and differentiates into plasma cells. These plasma cells are essentially antibody factories, producing and secreting vast quantities of antibody molecules into the blood and lymph. Antibodies do not kill pathogens directly, but instead bind to their antigens and tag them for destruction by other components of the immune system, such as phagocytes. T cells are responsible for cell-mediated immunity. There are several types of T cells, including helper T cells, which act as coordinators of the entire adaptive response by activating B cells and other T cells, and cytotoxic T cells, which are the direct killers. Cytotoxic T cells patrol the body, examining the surface of all cells. If they identify a body cell that is displaying foreign antigens on its surface, such as a cell infected with a virus, they will bind to it and release toxic chemicals that induce the infected cell to destroy itself, a process known as apoptosis. Supporting the cellular army is the lymphatic system, a network of vessels and organs that is intimately linked with both the circulatory and immune systems. The lymphatic vessels collect excess fluid, known as lymph, that has leaked from the blood capillaries into the tissues and return it to the bloodstream, thus playing a crucial role in maintaining the body's fluid balance. As the lymph flows through these vessels, it passes through small bean-shaped structures called lymph nodes. The lymph nodes are packed with lymphocytes and macrophages and act as biological filters. As the lymph percolates through a node, these immune cells survey it for the presence of any foreign antigens or pathogens. If an infection is detected, the lymphocytes within the node will rapidly proliferate, mounting an immune response and causing the node to become swollen and tender. The lymphatic system also includes organs such as the spleen, which filters the blood, and the thymus, the site where T cells mature. This entire integrated defense network, from the physical barrier of the skin to the specific memory of the lymphocytes, provides the cat with a robust and resilient system for maintaining its internal integrity in a world teeming with microscopic threats. The journey of an individual life, from the moment of conception to the quiet conclusion of old age, is itself a physiological process, a programmed and progressive unfolding of growth, maturation, and eventual senescence. The life cycle of the cat can be viewed as a series of distinct physiological stages, each with its own unique characteristics, challenges, and metabolic priorities. The journey begins with the neonatal period, the first few weeks of life. A newborn kitten is an altricial creature, born relatively undeveloped, with its eyes and ears sealed shut and completely dependent on its mother for warmth, nutrition, and stimulation. Its physiological systems are still immature. It has a limited ability to thermoregulate and must rely on huddling with its mother and littermates to maintain its body temperature. Its immune system is not yet fully functional, and it receives crucial passive immunity through the mother's first milk, the colostrum, which is rich in maternal antibodies. This period of intense dependency is followed by a phase of rapid growth and development. The kitten's nervous system matures quickly. Its senses come online and it begins to explore its environment. This is a period of enormous metabolic demand, fueled by the rich, high-fat, high-protein content of its mother's milk. The skeletal system is growing rapidly, and the processes of play and social interaction with its littermates are crucial for the proper development of its motor skills, coordination, and social behaviors. This juvenile period is a time of profound physiological and neurological programming, laying the foundation for the adult animal. As the cat approaches adolescence and sexual maturity, a cascade of hormonal changes, driven by the endocrine system, transforms its body and behavior. It reaches its full adult size and skeletal structure, and the reproductive system becomes fully functional. The adult stage is the longest period of a cat's life and is characterized by a state of relative physiological stability. The primary metabolic focus shifts from growth to maintenance. The body's systems are geared towards the day-to-day business of survival, acquiring food, maintaining territory, and for unneutered animals engaging in reproductive activities. The homeostatic mechanisms are at their peak efficiency, and the immune system has had time to build a library of immunological memories from exposure to various pathogens. As the cat moves into its senior years, typically considered to be from around 10 years of age onwards, the process of senescence begins. This is not a disease, but a natural, gradual decline in the functional efficiency of the body's various systems. The metabolic rate tends to decrease, which can predispose the animal to weight gain if its diet is not adjusted. The senses, particularly hearing and vision, may begin to decline. The cartilage in the joints can start to wear down, leading to osteoarthritis and a reduction in mobility and agility. The efficiency of the digestive system may decrease, and the kidneys, which have worked tirelessly throughout the cat's life to filter waste and conserve water, may begin to show signs of reduced function. The immune system can also become less robust, a phenomenon known as immunosenescence, making the older cat more susceptible to infections. This final stage of life is a slow, quiet unwinding of the complex physiological machinery that has sustained the animal for so long. It is a testament to the finite nature of this intricate biological engine, a gradual and peaceful return to simplicity after a lifetime of predatory perfection. And so, our quiet exploration of this remarkable creature comes to a close. We have journeyed through the intricate landscapes of its internal world, from the evolutionary blueprint that defines its purpose, through the silent, humming machinery of its cells and organ systems. We have seen the world through its light-gathering eyes, listened with its sound-funneling ears, and felt the whisper of air on its sensitive whiskers. There is no need to hold on to these details now. The purpose of this journey was not to memorize, but simply to provide a place for your mind to rest, to focus on the calm, logical order of a living system. Allow the complexity to fade, leaving behind only a sense of quiet appreciation. The steady rhythm of the information has served its purpose. You can now let go of the structure, let go of the words, let your own internal systems take over, those quiet, autonomic processes that govern your own rest. Your heart is beating steadily. Your breathing is slow and deep. There is nothing more to analyze, nothing more to learn tonight. There is only the peaceful, gentle pull of sleep. Allow yourself to drift on this quiet current into the deep and restorative calm that awaits you. Allow your thoughts to dissolve like sugar in water, leaving only stillness behind. It is time to rest now. It is time to sleep. Good night.